Hello, my name is Gabriella Caspi, and today I'll be talking about lipoic acid binding sites found in HSMVT. So, what are transport proteins? So, the transport protein is a transmembrane protein that spans the width of the lipid bilayer. So, the lipid bilayer is represented by the two gray lines, and the protein is the orange circle in the middle. Basically, what happens is a transport protein does exactly what the name specifies. It transports something, and that something is usually substrate. So the substrate is represented by the pink dots on top, which is on the outside of the cell, and they're transported inside the cell against some sort of gradient. Now, the substrate attaches to the protein at a specific spot called the binding site. And this binding site basically has the same idea that a lock and key has as pictured at the bottom of the slide. So the substrate is the key and the protein is the lock. So you can't have a random key open a random door. You need to have a specific key open a specific door, otherwise it's not going to work. Same idea applies for substrate and proteins. So relating to this study here, what is HSMVT? HSMVT is the human sodium multivitamin transporter, which basically means exactly what it says. It transports multivitamins. So in 1999, biotin and pantothenic acid were found to be substrates of HSMVT, and there was no evidence shown that there was a connection between lipoic acid and HSMVT. So that basically means that there were two known substrates of HSMVT up until now. Lipoic acid had never been connected with HSMVT in the slightest. What this study showed is something different. So I separated this into three parts. The big question, how we answered it, and what it means. Now I say we because I was actually a part of this experiment. I was an intern at Columbia University, and I basically got to take part in some protein purification and some cell culturing that was used in this experiment. So the big question, does HSMVT transport lipoic acid in addition to pantothenic acid and biotin? And how was this question answered? So basically what happened was we cultured 8 SF9 cells, which is a type of insect cells. And this basically after that, we expressed the HSMVT protein in these cells. So to express basically means that we use bacterial transformation to take the gene of interest, which is basically the gene that tells the cell to produce the protein HSMVT. We basically transfected the SF9 cells with this, so the SF9 cells would in turn produce the protein, which is HSMVT. So we did that, and then after that the HSMVT was then purified and isolated from the cell, and then binding studies were performed in a radioisotope lab. So from this, lipoic acid was found to bind to 2 places on HSMVT. So just a little more information on the lipoic acid. So basically what it means is that lipoic acid is a vitamin needed for normal biological function in humans. So this is a totally new function of HSMVT that was discovered, and it just gives us a little more information on what HSMVT actually does in biological functions in humans. So why is this new? So just to reiterate that point, HSMVT has never been associated with lipoic acid transport, and now we found that it has 2 binding sites. So most likely these 2 sites are allosteric regulators of the gating of HSMVT. So allosteric regulators, what does that mean? It's basically scientific jargon, which basically means it's the substrate, lipoic acid, is the key that opens another door on the protein that basically makes it open and close to put sodium in across the cell membrane. So it's basically as if you were opening a door with a key, and instead of opening the door where the key is, you open another door that would be connected to that, and that would let something else in. So basically what that means is lipoic acid is definitely necessary as basically the key for letting sodium in across the membrane. So that's very, very important. Also, in addition to this new information about HSMVT, a new method was used here. So basically, we obtained functional membrane protein, the HSMVT, in detergent solubilized form, which basically means a form that we could use to perform binding studies on. So as I mentioned earlier, in 1999, an article stated that there were only 2 substrates of HSMVT, which was biotin and pantothenic acid. Because they never had the functional membrane protein of HSMVT to do studies on, they could never really decide if there were other substrates that would be attached to it. So this method can be basically used in all different sorts of transport proteins, so not just HSMVT, but now binding studies can be performed on other transport proteins that might not have been able to in past years because of the lack of technology to do so. So this is also a great method that can definitely be used in the different transport proteins. So why is this significant? The information shown here creates a whole paradigm shift in the transport field. Basically, like I said, this new method can be used in different transport proteins, and now we know that transport proteins are the targets for many medications, and basically the more we know about them, the better medications that can be made, and also that can be prescribed. So basically knowing more information about them is going to give us a better sense of, what medication would be appropriate for a lack of vitamin X or a lack of substrate X or an over production of substrate X or whatever it may be. So this is definitely some great information that can be used with people who need specific medications. So on that topic, some medications targeting transport proteins, so you have a bunch. So you have probably one of the most famous is Prozac, which is one of the very heavily prescribed antidepressant. You also have amiloride, which is used to treat congenital heart failure and high blood pressure. And then you have other drugs that treat OCD, other mental illnesses, some that lower cholesterol, other that treat mental illnesses such as panic disorder, social anxiety disorder, and other antidepressants. So these are obviously very, very important drugs for many people who suffer from these diseases. So once again, knowing more about the transport proteins that the drugs target will lead to better creation of drugs and better prescription of drugs. So to sum it all up, there was novel binding of lipoic acid in 2 sites on HSMVT and the new method in isolating HSMVT. These are 2 new things that were discovered in this study. So the methodology as well as the function of the new substrate as an allosteric regulator of the gating in HSMVT can be translated over into other transport proteins. And like I said, the more information we have on transport proteins, the better medication we'll make. So that's all I have to say. Thank you very much for watching.